Hello, everybody, and thank you for joining here on Oncotelic's channel. We once again have a special guest for you guys. We have Dr. Anthony Maida, the Oncotelic's Chief Clinical Officer. Anthony, how are you holding up over there?
Very well. Very exciting time, but all is well.
Good, good. Glad to hear it. Yeah, 2022 is going fast. We're at the end of first month already. Surprising how fast things go. But can you tell us a bit more about what your role at Oncotelic as Chief Clinical Officer really entails?
Certainly. The position requires overall and oversight of all clinical functions, the planning of the clinical studies, the regulatory issues, dealing with all of the PIs and potential PIs, identifying the KOLs that are going to be doing the studies, working with CROs, and essentially coordinating all aspects of the conduct of the clinical trial to bring us either to the next inflection point or to an approval.
Okay, and how much experience do you have doing this? I imagine this is a very important job, very important to dot all your I's and cross your T's on.
I've been doing this for about 30 years. All of those 30 years have been in immuno-oncology. In fact, way before it was called immuno-oncology, and have overseen a number of clinical trials, initiated a number of clinical trials, working with the KOLs, SIVs, SQVs. And as you might expect, after 30 years, I have a huge Rolodex of physician scientists that are working in various indications, from pancreatic cancer, non-small cell lung cancer, Merkel cell cancers, very, very rare cancer, the gliomas, and it goes on and on.
Okay, yeah, I mean, you are an expert in immuno-oncology, and from there, can you highlight the reason why you liked Oncotelic and why you came on board from your experience perspective?
There's really no one reason. I've known Dr. True for a number of years. He really ushered in Abraxane into the clinic. He is absolute genius. There are a number of very good, hardworking people in this company. It is manageable. Obviously, the drug attracted to me, OT-101, which is an ODN, oligonucleotide, oligodeoxynucleotide, antisense. And I think that's where the science is going to be going next from antibodies. And then also the attention of the team on the inhibition of TGF-beta, which I think is huge. There is an enormous explosion in research in the inhibition of TGF-beta, not only as a monotherapy, but along with chemotherapy. And obviously where we're going is in combination with checkpoint inhibitors. And what we want to do is, to use the old hackneyed expression, we want to turn these cold tumors to hot tumors. Really overuse that expression. But anyway, the trials are in design exceedingly robust. And we're going to have accompanying those trials an extremely thorough biomarker study that is going to encompass all the clinical trials that we are going to be conducting over the next year to 2 years, measuring both pre and post changes to the tumor microenvironment.
So Doc, you convey a lot of the very high level stuff, but if you could break things down for your common layman, just to express the potential of Oncotelic, how would you do that? How would you best say that?
Well, I think over the last 10 years, you've seen a massive explosion in the use and approval of checkpoint inhibition and inhibitors. But when you really start looking at the benefit, only about 20-25% of those patients benefit from the use of checkpoint inhibition. So there is huge room for us to augment and improve the response to checkpoint inhibitors. Checkpoint inhibitors are here to stay. So now how do we build upon that? Do we include TGF beta, which I think is a very strong opportunity, but also maybe combining checkpoint inhibition with TGF beta along with certain chemotherapies. And those chemotherapies can essentially act as in situ vaccines. And essentially chemotherapy destroys tumor cell releases antigen. Those antigens are taken up by the dendritic cells, presented to T cells. You now have a anti-tumor response to T cells. But what can happen many times is those T cells migrate to the tumor. They will express gamma interferon. And the tumor cells will essentially respond to gamma interferon by upregulating PD-L1. And so PD-L1, once it's expressed, is a ligand for PD-1 and essentially shuts down the T cell. So what we want to do is use checkpoint inhibition to interrupt that dialogue, and then to use TGF beta to improve upon the response and the response of a number of immune cells that are infiltrating the T cell that are immunosuppressive. So it's a very complicated story, but quite frankly, it's also very simple. We know what we need to do, and we know how to work with the various drugs out there and checkpoint inhibitors, both anti-PD-1 and anti-PD-L1.
Yeah, Anthony, I think you're right. While it does seem complex, I feel like you expressed it as simple as you possibly could there, everybody. But if anyone has any other questions about it, they can obviously ask. We can get some answers out to them as soon as we can. So do you have any closing words, anything else you want people to know about yourself before we go?
About myself, no, I don't like ringing my own bell. But what I would like to do is impress upon the group that we are planning a number of clinical trials in non-small cell lung cancer, mesothelioma, pancreatic cancer, melanoma, gliomas, particularly DIPG, which is diffuse intrinsic pontine gliomas. Typically, that's a pediatric indication. Absolutely nothing for these kids. No therapy works. So the only therapy is palliation. But, and the palliation is in the form of radiotherapy. And now these kids really start losing cognitive function. So we're going to be conducting a number of clinical trials. And what we want to do is essentially move towards an agnostic approval, something like what Merck did with those patients that are MSI high or deficient in mismatch repair. And so every single study that we're conducting, we're going to get pre and post tissue biopsies. And then we're going to measure cytokines, RNA-seq, MDSCs, M1, M2 macrophages, various levels of TGF beta, pre and post. And it's really robust, very comprehensive to see if we can find the predictors of those patients that are going to respond and those that are not. And that's going to be through the changes in the expression of various biomarkers. So I'm really excited about all of these studies, and I'm really hopeful that we're going to be able to change the course of disease.
Yeah, that's a great thing to reach for there. On just to wrap it up, what's about the milestones of these studies, and how long people can hear more information on them, like when they're going to finish and so on?
Well, a number of these studies we're going to be working with a very large pharma. We can't release their name right now. Some of the studies we're getting drug. Some of the studies we may be getting drug and funding. The first study, mesothelioma, the initial concept has been approved by this pharma. We've drafted the protocol. And so I expect that clinical trial just to be safe, say 3 to 6 months before we start enrolling, and then about 2 years to enroll. Piggyback on that, we're going to be doing the non-small cell lung cancer. Protocol should be very much the same as the MPM trial, the mesothelioma trial. And then all the other studies will start probably by the end of this year.
All right. Well,
So very aggressive, very aggressive.
Yeah, working fast. And I mean, it's like I had talked about with lots of the other people I was talking to Amit. You guys are working in an industry where time matters. Getting stuff done leads to things getting to people potentially.
Absolutely.
So on.
Absolutely. A huge sense of urgency to get these studies moving.
Well, Doc, I think you did a really good job of shedding light on your role, shedding some insight on things. I know a lot of people don't quite understand the ins and outs of it, but I think you did a great job of breaking it down for everybody. But again,
well, anybody can talk to me and ask me. You know, obviously there are FD regulations, so we have to be careful what we disclose. But I'm more than happy to answer the clinical and scientific basis for what we're doing.
Wonderful. Well, you guys heard the man. If you have any questions at all, don't be afraid to reach out. We will happily dive into them. But for now, stay tuned for another video coming soon. Anthony, thank you so much for your time today.
Thank you very much. Have a lovely day.
You as well.